Today, I'd like to talk to you about two important reactions, the reaction of photosynthesis and the reaction of cellular respiration. And both of these reactions have to deal with energy, and energy is the ability to do work. For all living organisms, the ultimate source for all energy is the sun. And it is the process of photosynthesis that captures sunlight energy and stores that energy by making glucose. So photosynthesis captures sunlight energy and stores it by making the bonds of glucose. So one important concept when it comes to energy is that when we make bonds, when we make chemical bonds, we store energy. And that's the primary focus of photosynthesis, that we are making bonds by making glucose, and in doing so, we are storing the energy from sunlight. The second reaction, cellular respiration, that is the reaction that breaks the bonds of glucose, therefore releasing that energy. Which then comes to our second important concept, that when we break bonds, we release energy. And that's the primary focus of cellular respiration, that we are breaking the bonds of glucose, therefore releasing energy. Yes, this is all in the name of science.
Sometimes it works, sometimes it doesn't.
Oh, this will work. This better work.
His life is flashing before his eyes.
Good luck.
Oh my god.
So, the light and the heat that's coming off, that's the energy from the chemical bonds that are released. So we'll begin first by looking at photosynthesis. And the reaction for photosynthesis is carbon dioxide plus water yields glucose plus oxygen. So if I take a look at this reaction, the first thing I need to have is sunlight and chlorophyll. To look at chromatography of the pigments in, say, spinach, we can take some baby spinach leaves and rip them up, add it to our mortar and pestle here, and then we can add a small amount of acetone. And acetone is going to help extract out the pigment that is in the spinach leaves. And then I'll begin grinding as such until I get some green liquid. That's my acetone with the pigments now extracted. I can then take a thin layer chromatography plate, as you see here, and spot it with that pigment. So I'll begin by drawing a line in pencil. And then taking a capillary tube, I'll dip into some of my liquid and then touch my plate like such. I'm going to allow that to dry, and then I'm going to spot it again, say 5 or 6 times, until I have a nice dark spot on my plate. I will then add this to some chromatography solvent. As it sits in the solvent, the pigments will separate according to size. The different color bands that you see corresponds to the different pigments that are found in spinach. We can take a piece of leaf here. Here I have a jade leaf, and very carefully we can cut a thin section of it. Push it onto a slide. Add a drop of water. And then take a cover slip. Add it on. Press gently. And then use the microscope to look for chloroplasts. We can see some plant cells. The borders that you see here are the cell walls, and the green dots that you can see within the cells are the chloroplasts that contain the green chlorophyll. The process of photosynthesis can be divided up into 2 separate reactions. The first part is called the light-dependent reaction. So here we have chlorophyll in our chloroplast, and sunlight is going to hit the chlorophyll. As a result, an electron is going to come off of chlorophyll, where it will then enter into the electron transport chain. That electron will get passed from carrier to carrier till we get to the final electron acceptor called NADP+, and when it accepts that electron, it becomes NADPH. While this electron has been passing through the electron transport chain, protons are going to pass across the transport chain and then fall down their concentration gradient through a molecule called ATP synthase. As the protons pass through ATP synthase, it generates the energy for ADP to gain a phosphate group to yield ATP. Now I'm positive that the chlorophyll has lost an electron, get the joke, and to regain the electron, we take one from water, which yields oxygen. So if we look at our reaction, the water to the oxygen, that is the result of the light-dependent reaction. The second part is the light-independent reaction, otherwise known as the dark reaction or the Calvin-Benson cycle. Here, carbon dioxide is going to be fixed into glucose, and what drives this cycle is the NADPH and the ATP that we generated during the light-dependent reaction. So if I look again at my reaction for photosynthesis, we see that this portion of the reaction, the carbon dioxide to the glucose, is the result of the Calvin-Benson cycle. And so we can observe the production or the result of glucose production by examining for starch granules that are found in banana smears. we can look for starch granules in a smear of banana. So to detect starch, we use Lugol's solution, which contains iodine. And iodine will react with starch and turn from a brownish color to a bluish purplish black color. So I'll take some iodine and just add it onto my banana smear. Let that sit for a second. Take some deionized water and rinse gently. Tap off the excess. Take a cover slip, add it at an angle, drop, press gently, and then using the microscope we can look for starch granules. So here we can see some of those starch granules. They've turned a bluish purplish black color, and you can see the production of starch as a result of photosynthesis. The oxygen we can also observe by using a sensor. We can measure the oxygen concentration from the plants in the tank that you see here. One tank has a light bulb being shined on it, and the other tank is underneath the black bag that you see. Here is a graph of the data collected from that sensor, and you can see that the concentration of oxygen is increasing in the tank with the light, while the concentration of oxygen is decreasing in the tank that is in the dark. It is increasing in the light because of the process of photosynthesis occurring because sunlight is present. So now we'll take a look at aerobic, which means using oxygen, so aerobic cellular respiration. So our reaction is glucose plus oxygen yields carbon dioxide plus water and ATP. And if you notice something, you'll notice that this reaction, cellular respiration, is the opposite of photosynthesis. Photosynthesis reaction was carbon dioxide plus water yields glucose plus oxygen. So if you know the reaction of one, then you know the reaction of the other. Respiration takes place in 4 steps. The first step is glycolysis, which means sugar. Glyco means sugar, and lysis means breaking. So we are going to break down glucose. And this occurs in the cytoplasm of the cell. So here we have glucose, and we use 2 molecules of ATP to break glucose into 2 molecules of pyruvate. Glucose is a 6-carbon sugar, and pyruvate is a 3-carbon molecule. We also generate NADH, an electron carrier, and we make 4 ATP molecules. But because I use 2 of them, my net is going to be 2. The second step is pyruvate oxidation. This occurs in the mitochondria of a eukaryotic cell. And here we take the 2 pyruvates that we made from glycolysis, and they are converted into 2 molecules of acetyl coenzyme A. We also generate, I forgot to write it in, some NADH, those electron carriers, and carbon dioxide, one of the products of respiration. The third step is the Krebs cycle, also known as the citric acid cycle or the TCA cycle. This also occurs in the mitochondria of a eukaryotic cell. And here we have the two acetyl coenzyme A's that we made from pyruvate oxidation. And when they enter the cycle, we're going to generate carbon dioxide, again, one of the products of respiration, our electron carrier NADH, and another electron carrier called FADH2, and 2 molecules of ATP. So if we look, we see that the glucose to carbon dioxide step is the result of step A, glycolysis, step B, pyruvate oxidation, and step C, the Krebs cycle. So A, B, and C are all this portion of the reaction. The fourth step is the electron transport chain, which also takes place in the mitochondria. And NADH and FADH2, which came from the pyruvate oxidation, the glycolysis, and the Krebs cycle, all deliver their electron to the electron transport chain, where the electron will get passed from carrier to carrier till we get to the last electron acceptor, oxygen, and when oxygen gets that electron, we form water. While the electron is being passed, protons will move across the electron transport chain and then fall down their concentration gradient through ATP synthase, and that will provide the energy for ADP to add a phosphate group to yield ATP. So looking back at my reaction, the oxygen to the water portion, that is the result of the fourth step, part D, the electron transport chain. We can observe this reaction, so we can look at the products of respiration, specifically carbon dioxide, by preparing a culture of yeast, sugar, and water, and attaching either a balloon or using a sensor. Here I have a culture of yeast where I've taken some dry active yeast from the local supermarket, added some water and some sugar, and then put a balloon on top. And you can see the balloon has begun to expand because through the process of respiration, carbon dioxide is being produced. And I can verify that it is carbon dioxide by using a carbon dioxide sensor here. So I will remove the balloon, hold my sensor over the tube, and then when I press play, you can see that it is detecting carbon dioxide being released from my yeast culture. When ATP is incubated with luciferin and luciferase, glowing occurs, and that's how firefly tails glow in the dark. Here's a vial of firefly tails which have luciferin and luciferase. So if I have a solution of ATP, I can add that solution to my tails. And then after a few moments in the dark, we will see some glowing, indicating the presence of ATP. Respiration that occurs in the absence of oxygen is referred to as anaerobic respiration, and a special type of anaerobic respiration is called fermentation. So fermentation is respiration in the absence of oxygen. And in the process of fermentation, only glycolysis occurs. And so different organisms that can ferment will produce different end products as a result of this fermentation. For yeast, that includes carbon dioxide and alcohol, which we can measure in the lab. And for human muscle cells, that results in the buildup of lactic acid, where your muscles are performing respiration but not getting enough oxygen. So the muscle cells begin to produce this fermentation product of lactic acid, and that's what gives you your muscle cramps after a long period of exercising. You can measure the ethanol production from yeast that is performing fermentation by either using an ethanol sensor. So I can open up my culture, stick in my sensor, and then you can see that the percent of ethanol is increasing. Alternatively, I can use a fermentation tube that you see here, and if you look at the top, you can see the formation of bubbles, which indicates carbon dioxide production as this culture is performing fermentation.